Recently, a rapid surge in gas prices, destructive oil spills, and increasing climate concerns have prompted a strong effort by politicians and scientists to develop sustainable fuel sources. One major clean energy initiative is the Build Back Better infrastructure bill, which includes a $1 billion investment in biofuels. Biofuels are produced from biomass, like algae, and in addition to being carbon neutral, they have many unique benefits. I'm Ashwin.
And I'm Rohan, and we developed Algaeorithm to support the development of algae biofuels.
Upon opening Algaeorithm, you're prompted to either enter image URLs or upload images from your device. You also have the option to specify whether or not your data is time sensitive and whether or not your images all have the same depth. After submitting images, you first see a statistical overview of your image set, along with graphs to visualize distribution and model growth for time-sensitive data. You can also view your image data in table form and download it as a CSV file. To see specific images, you can click on the corresponding row in the table or click on the Image Analysis tab at the bottom. In the Image Analysis tab, you can see how Algaeorithm interpreted the image and the corresponding cell count and concentration. After the user uploads a photo, we use multiple image processing techniques to accurately count cells and calculate concentrations. First, we use binary thresholding, which converts the image into its foreground and background elements. And then, contour detection to find isolated elements in the binary image. Figuring out how to decipher cell clumps was a big challenge. We solved this by using local peak maxima calculations to separate clumps into circular elements, and then comparing the radii and centers of the circles to distinguish between multiple distinct cells and one elongated cell. To build our algorithm, we used packages from Python for image processing, statistical calculations, and graph creation. We turned the algorithm into a web application using the Python framework Flask to make it accessible from any internet-connected device. The front end was made with HTML, CSS, and JavaScript, and the entire application is deployed through Heroku.
Algaeorithm was able to measure cell count and concentration with over 90% accuracy for Chlamydomonas algae and can provide information about the images in a matter of seconds that would take a human minutes or even hours. With time-sensitive data, we're able to model long-term biomass accumulation. All of these features make Algaeorithm useful not only for scientists in the lab, but also students in the classroom, as it provides them with research-caliber information.
The Algaeorithm app bridges a much-needed gap between experimental observations and computational biology, but also provides an exciting new opportunities for add-on capabilities.
We hope Algaeorithm will assist the next generation in transitioning to a cleaner world.